Hello, so we're going to start talking about the control of muscle contraction here. My name is Dr. Ryan Miskowitz. So, controlling muscle contraction really has everything to do with the nervous system. Okay, um, nerves are the electrical system of the body, and in the case where we're concerned about muscles, muscles are going to be the mechanical. So, if you don't have electricity to, say, an electric motor, the motor doesn't do anything. And that's roughly what happens. It's the relationship between muscles and nerves. Okay, so nervous system. Um, Starting at the top, and we don't get too much into this in XPhys, but just to briefly mention it, your brain and central nervous system. Your brain is the core of the central nervous system, as is the spinal cord. Um, Sometimes we just say CNS for this. All right, so the brain and the spinal column, that's the control, if you will. From there we have the peripheral nervous system, PNS, sometimes referred to as. Peripheral nervous system is divided into two main groups, those being the sympathetic nervous system and the parasympathetic nervous system. And as we go through not just this unit, but the rest of the semester, and as you guys are out working in the field, um, it's important to know the actions of these two nervous system groups because they come up. So, sympathetic nervous system, or the SNS, essentially functions as the gas pedal. It's the go, go, go. All right, so when your sympathetic nervous system sends input, in general, it's telling your body to go faster, contract harder, that sort of thing, okay? It wants action. The parasympathetic nervous system works as the opposite, okay? It's if the sympathetic nervous system is the gas, the parasympathetic is the brakes, okay? So the parasympathetic acts to calm things down, slow down rhythms of the body, reduce contractility, that sort of deal. Okay, so coming off of the spine and out of the brainstem, you're going to have these sympathetic and parasympathetic nervous system neurons, and those are going to meet with motor neurons. Okay, and a motor neuron is actually the nerve that interacts with the muscle fiber. Right. So more of an overview here. We have 12 cranial nerves which connect to the brain, 31 spinal nerves which connect with the spinal cord. All right. The sensory division, which truth be told we're really not going to talk about too much at all. The sensory division, it serves to give feedback to the brain and the spinal column. It gives you information. From there, your brain and spinal column make decisions. Okay, so the motor division. The motor division is where those determinations of how to move are formed. And an autonomic nervous system, that's control of involuntary functions such as breathing or the heart rate or things like that. Okay. So this is what a neuron looks like. All right. So roughly we have our cell body here. Okay. So you had from within that the nucleus here, and then these guys out here are called the dendrites. Okay. Now at the dendrite, that's where this nerve actually gets its signal to do something. Okay. So you'll have sympathetic, and we'll see this later in a different frame, but you'll have, say, sympathetic nervous system neurons here, maybe some parasympathetic ones, and this neuron right here is going to receive a certain amount of signal from the sympathetic and the parasympathetic. That will be interpreted by the nucleus as to what the response should be, and depending on what it deems an appropriate response, you'll have some sort of an action. Okay, so not getting too far ahead of ourselves, that's essentially what happens at the nerve cell body up here. All right, any sort of an action is going to be propagated down this axon. Okay, Now in mammals, we have these things, myelinated sheaths. So the myelinated sheaths kind of help to speed up the speed of contraction down the axon. That's pretty much all they're doing. And then at the end, once a signal goes down the axon, it reaches the end here to the synaptic knob, or you can call it the terminal. You know, there's a couple terms for it, but anyway, from here it's going to send the signal down the stream to the next place. Okay, so if we're talking about connecting to a muscle, well then it's going to tell the muscle to, say, contract. Okay. Hold on. Okay, so a term you'll hear often from me saying it is motor unit. Okay, so a motor unit is comprised of the muscle fibers. Okay, the muscle fibers, say this is a motor unit right here. This would be one motor unit. Okay, so the motor unit is this neuron that's coming off of some other peripheral nerves higher up the chain. Okay, so this neuron right here and the fibers that it's connecting with. Okay, you see it's, in this case, it's innervating 3 fibers, right? 1, 2, and 3. Okay, so this would be one motor unit. Okay, and this one motor unit is one neuron and the 3 muscle fibers that it innervates, or it meets up with. Essentially the ones it's in control of. Okay, so motor units, they kind of vary greatly as far as the number of cells which are assigned to a single, the number of muscle fibers assigned to a single neuron. You see here as little as 4 to over 1,000 muscle fibers can be connected to a single motor neuron. Okay, now the relevance of this, what you need to be thinking about here is why. Okay, as we're going through, I want you to understand the why of things. Okay, so if I have one motor neuron that intercepts or innervates 1,000 fibers or more, it probably means that that motor neuron is not involved in a great deal of highly coordinated activity, right? Because every time it fires, it's going to contract 1,000 fibers, right? 1,000 fibers are going to produce a lot of force, but they won't be very good at fine motor tasks, say like trying to write with your pencil or moving your eyes around. You know, those are very fine movements. And if you have 1,000 muscle fibers all contract at the same time, you're going to get very kind of herky-jerky, if you will, uncoordinated movement. Okay, so the more coordination that's required, you get a smaller ratio of muscle fibers to motor neurons. Okay, so that's a characteristic we'll touch on a little bit later. All right, so that's the basic functional unit of a muscle, though, is the motor unit. Okay. All right, so impulse movement. So how does that neuron talk to, if you will, the muscle fiber? Well, the simple answer is through action potentials. Okay, an action potential is a depolarization across the neuron and muscle, and basically what happens is you have this neuron, and it's got a certain amount of ions on the outside and on the inside, and it holds typically a fairly negative voltage. Okay, so it's negatively charged, and once enough signal goes along, the membrane increases its charge. It becomes more positive. Once it becomes positive enough, it triggers a threshold. It hits a threshold of depolarization, and that causes an action potential to fire across the neuron or propagate across the neuron, as is often said. Okay, the movement of this action potential across a neuron is accomplished through things called voltage-gated channels. Okay, Voltage-gated channels allow ions to move across the neuron due to changes in voltage, okay? And we'll see this a bit later, but the other kind of channel you're going to get is chemically gated. A chemically gated channel, those are typically the points where you're going to have the start of an action potential, okay? So they're opened through actual chemical interactions. Okay, an example of this is going to be the motor end plate, and we'll see this a bit more later. All right, so this is kind of a little graphical representation of um, the cell membrane and the different kind of ions we have. Okay, so here, right here, this is a chlorine ion. We have sodium ions. We have potassium ions, and this A with the negative superscript, that just stands for negatively charged ions or anions. Okay, so essentially inside of the cell or in the intracellular fluid, you have a mixture of different ions which are negatively charged, and for simplicity we just write this A negative. Don't get too concerned with it. All right, so we have intracellular fluid here, and then we have extracellular fluid here. So this is outside the cell, okay, and this is inside the cell, right? Okay, so we're going to start here, and we're going to move along. I'm going to show you the process of a depolarization. It's important you know this stuff. And I have an animation set up for a little later in this video, and hopefully it'll bring it all together for you guys. So just stay with me. Okay, so let's take this. This is a nerve cell here. Okay, this is a nerve cell, and we're going to start the process of moving an action potential across this nerve cell. Okay, so first off, we've got, this is the voltage right here. Okay, so pay attention to the voltage as we go. This little red dot is going to kind of show you where we're at in the process of depolarization and then repolarization afterwards. Okay, so we're starting here at resting potential. All right. So this is what it looks like when a nerve is at rest, or a muscle. Okay, the outer part of a muscle cell, they roughly work the same. So, but in this case, a nerve. When this nerve is at rest, we have these blue. They're just arbitrarily blue, so don't get bogged down in this. But we have our blue sodium ions on the outside, and our brown potassium ions on the inside. Okay. All right. So you have a sodium channel here, and we have a potassium channel right here. And then we have chlorine on the outside, and just kind of unnamed anions floating on the inside. Okay, so next step. We're starting to have a change in membrane potential, okay? We're starting to have some facilitation. Facilitation is just what happens when a cell starts to become more positively charged, okay? When you start to have this charge moving more toward threshold. Once it hits threshold, it's said to be excited, but before that, it's said to be facilitated. We'll talk more about this in a later unit. But anyway, we have our sodium ions, and the way that this neuron is going to become more positively charged is these sodium ions are going to start going through the sodium channel. Okay, you can see some of them have already come in here, right? All right, so we have all our sodium ions. They're going into this sodium channel. Okay, now they have a positive charge. So as this cell starts gaining more positively charged sodiums, we're getting more positivity on the inside, right? Okay, so if you're getting more positive on the inside, well, the inside starts to become more positively charged. Okay, and if we're losing positivity on the outside, it's starting to become more negatively charged, right? Makes sense? Okay. So moving along, we've reached potential. Okay, we've reached threshold. Once we got enough of these sodium ions inside, we don't have any more outside, or there would be some, but just for this animation, we're going to say they're all out. Okay, so the sodium ions have gone into the cell. The cell has become positively charged. Okay, outside is now negatively charged. This neuron is said to be excited. Okay, so we've reached this very high level of positivity, and this is going to start going across the neuron. Okay, it's gonna spread itself. But for now, this is what it looks like when it's at its most positive. We have sodiums on the inside and potassium, so all of these are positively charged. Okay, and so forth. I don't need to do all of them. Okay, so when we need to repolarize, which we do, if a nerve never repolarized, well then, in the case of, say, a motor unit, the motor neuron, the nerve that intersects with that muscle fiber, it would keep telling that muscle to contract, and you'd have a rigor, a cramping, okay, where that muscle just would stay contracted and never release. So you don't want that. It's not very good for motion, right? So it's important that after you depolarize, you can repolarize and get back to where you were to begin with, and that way you can have another contraction. Okay, so the process of repolarization starts with these potassium ions going out through the potassium channel. Okay, so the potassium, both these channels do not require ATP. Okay, they're passive. All right, so it's important to know where you use ATP, and we'll talk about it more as we go. So potassium starts to flood out. Once again, potassium is positively charged. Okay, so if you're gaining positively charged cells, or rather ions on the outside of the cell, then you start to get a rush of positivity. Okay, so outside starts becoming more positive, inside starts becoming more negative. Okay, and that's seen right here, right? So we're moving down. The voltage is starting to become more negative. We're starting to become negatively charged. Okay, we're moving back down toward rest. Okay, so the next slide here, we have, just for the sake of simplicity, all of our potassium is on the outside, all of our sodium is still on the inside. Great. Now we have moved down, we become more negative, we're getting closer to resting, but we're not there yet. Okay, the final step in this process, after you get sodium in and then potassium out, is to exchange them back to where they came from. Okay, back to where they started. And that's accomplished through this thing called sodium-potassium pump. Okay, so sodium-potassium pump actually pumps potassium in and sodium's out. Okay, so that's its job. This requires ATP. Okay, so for this thing to work, you're going to burn up ATP. All right. Now, this is a major site of ATP usage in the body, just regulating membrane potentials. Okay, so as we go forward and we're talking about where is ATP used and how is it produced and the relevance of that, just remember that the sodium-potassium pump is one of the major sites where you use ATP in the body, where when you're running and you're starting to fatigue or tire out, to a certain extent, just changing the membrane potentials of your muscles and the nerves is causing that to happen. Okay, this is one of the drains on your energy sources, the body. Okay, so moving along, sodium-potassium pump is exchanging sodium and potassium back to where they started from. You can see here resting potential. Well, sorry, the potential is getting lower. Okay, we're getting back toward rest. And this process continues, and as it continues, you see the charge becomes more and more negative, and eventually we're back to where we started. Okay, so sodium's back outside, potassium's back inside, and we are at resting potential. Okay, that's where we want to be. At this point, the neuron is now ready to depolarize again. Okay, and that is the depolarization, repolarization cycle. Okay, just in a very slow kind of format. Now let's think about this a little bit more globally. Okay, I gave you the nuts and bolts of how it works, but think about this across an entire neuron. Okay, so this is our neuron right here. What you see is this depolarization occurs regionally across the neuron. It's not the whole thing, okay? It's not the entire neuron going at the same time, right? Instead, you have this segment right here depolarizing, right? And, the process we just talked about, that's what's happening. When this part starts to depolarize, it's going to cause this one to depolarize, and then this one, and then this one, and then this one, and then this one. Now, on the back end, repolarization is happening kind of at the same pace, so we're going to have repolarization kind of following suit, okay, afterwards, right? So they follow one another. And this is just the same thing. If you look at this, you have this part is now repolarizing, and it's just moving the signal down the line, right? It's a bit like, I like to think about this as the wave at a stadium. Okay, if you go to a football game or a baseball game or what have you, everyone in the stadium doesn't stand up and throw their hands in the air at the same time, right? You wait for the person who was next to you to do it, then you stand up, and then the person who was sitting next to you stands up. And you've seen the wave before, so we don't really need to go into the nuts and bolts of that. But that's how it works. Okay, you get this spreading of depolarization and repolarization afterward down the line, one segment after the other. Okay, now onto these myelinated sheaths, okay, or what a myelinated nerve is versus a normal nerve, okay? So in a myelinated nerve, you have these myelin sheaths right here, and the purpose of this is instead of having to depolarize the entire axon to send the signal, these neurons kind of go from one little break here, which you call it a node of Ranvier. It's French. Anyway, this signal will be sent from here, skips that segment, goes to here, skips this segment, goes to there, and instead of having to depolarize all of this, we get to skip, right? So it allows you to convey that signal at a faster rate. Okay, so some more terms we should know. The neuromuscular junction. So a neuromuscular junction is the place where the neuron and the muscle fiber interact. Okay, that's where the innervation is occurring. A motor axon terminal is the ending point of a motor neuron. Okay, so a neuron which is going to be interacting with a muscle fiber. This is the point where it actually connects with the muscle, and this is where you have storage of neurotransmitters in the form of acetylcholine. Okay, acetylcholine is the neurotransmitter that tells muscle fibers to depolarize. Okay, if you will, they send the signal for the muscle to contract. Okay. Neurotransmitters in general, the purpose of a neurotransmitter is to convey a nerve impulse, convey a signal that the nerve was sending across a gap, okay? Because as we've talked about the depolarization of a neuron, and the importance of that sends a signal from one place to another, but it only sends it through its own cell, okay? That neuron is unable to send the signal, to translate that signal directly to whatever it touches. So instead, you'll have that neuron get excited. It sends the action potential down its axon. Once it hits the axon terminal, it sends out neurotransmitters. Those neurotransmitters then interface with whatever that neuron was talking to, and then it sends the signal down that and on down the line. Okay, so acetylcholine, as I stated before, is the actual neurotransmitter responsible for conveying signals from motor neurons to muscle fibers. Okay, and this is what that looks like. So here we have our motor neuron. Okay, so we had an action potential, we had an AP here, it went down. Now, this neuron, it looks like it's just directly touching the fiber. And to a certain extent it is connected to the fiber, but it's not able to just directly say, okay, I depolarize, now you depolarize. Instead, what you need to have are these acetylcholine molecules come out and be released, and they go across this thing called the synaptic cleft. Okay, so the synaptic cleft is this kind of gap, if you will, all right, this gap, and the neurotransmitters cross that gap. Okay, so they go out of the neuron, they cross the gap, and then they bind up in these end plates, these motor end plates. Okay, and once they bind here, they open chemically gated channels in the muscle fiber, and that chemically gated opening of sodium channels causes the depolarization across the muscle fiber. Okay, so these chemically gated channels essentially start the depolarization of a fiber, muscle fiber. There's a thing called an all or nothing principle, which should be stated. Okay, the all or nothing principle, as it relates to nerves, states that every muscle fiber that a motor neuron innervates will contract at the same time, regardless of how many fibers there are. So if you have 3 fibers connected to a motor neuron or 3,000 fibers connecting to a motor neuron, once that action potential is sent, once you have propagation of that signal across the motor neuron, everything that it touches is going to contract simultaneously. Okay, and a single action potential, one single propagation of signal, results in what we call a twitch, a single twitch. That's your muscle fiber contracting one time very rapidly, just kind of a one and done kind of deal. Moving on from here, we'll see about modulation of force, and you'll see that multiple twitches can summate and they can build on each other to form stronger contractions and that sort of thing. But for now, let's just leave it with this, and I'm going to give you guys a quick animation to watch. Hopefully it helps to bring everything together.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside of the cell.
Neurons control basically when the muscle turns on and off, but within the muscle, we have to start thinking about, what kind of the mechanisms for controlling contraction, and that's mostly going to be through calcium. Okay, so when a muscle depolarizes, the sarcoplasmic reticulum releases calcium to the sarcoplasm. Okay, calcium binds with troponin, and the cross-bridge cycle commences. Right, we talked about the cross-bridge cycle in an earlier video. Okay, so the muscle doesn't stop contracting. Okay, those cross-bridge don't stop cycling until calcium is removed from the troponin and from the sarcoplasm by extension. Okay, because if there's calcium in the sarcoplasm, the troponin is going to try to grab it up. Okay, so let's talk about calcium removal. Okay, so the sarcoplasmic reticulum, it releases calcium, but it also has pumps called calcium pumps to retrieve that calcium. All right, and the way I like to illustrate this is it simply works on a system of affinities. Okay, the affinity of calcium. So calcium's desire to go one place or another is determined by its affinity for that place. Okay, so troponin C, all right, it has a strong affinity for troponin C. So when calcium is in the sarcoplasm, it'll try to go and bind with the troponin C. But it has much more affinity for calcium pumps. Okay, and this is kind of what it looks like. Okay, say you are calcium, it's about lunchtime and you're hungry. You can either have this or this. All right, they're both cheeseburgers. They will both make you happy, but I think we know where we would go. We would land on this one. Okay, so calcium pumps are just more appealing. So calcium will, when available, go to the calcium pump instead of troponin C. This allows for depolarization, or rather relaxation of that muscle fiber, the cross-bridge cycle to stop. Okay. Now, calcium pumps. Calcium pumps are not simply turned on, say, like when you have a contraction. Calcium pumps are kind of always going. Okay, they're kind of always working. The example I like to give for this is if you think of your sink. Okay, you have a drain in the sink. Okay, drain, and you also have a faucet. Okay, so the water is the calcium in this instance. The calcium pumps are always trying to pull calcium out of the sarcoplasm. Okay, and the sarcoplasmic reticulum only releases calcium when it gets a depolarization across the muscle, when it gets a signal to do so. All right. Now, the small caveat here, when you're thinking about this drain right here, all right, I want you to think about it as being somewhat stopped up. You know, let's say uh, you're not so good at using a garbage disposal. There's a little bit of food in the drain, and it's backing up a little bit. It's draining, but it's not draining at the rate that the water's coming in. Okay, there's a little bit of a backup if you let the water run. Okay, um, the fact that calcium pumps cannot remove calcium at as fast of a rate as the sarcoplasmic reticulum can release it is highly, highly important as we move along and we start talking about strength of contraction. Okay, so just think about this in these kind of abstract terms for now. Um, Calcium is released, calcium pumps are always trying to pull that calcium back, but their rate of removal does not match the rate of release if the sarcoplasmic reticulum is actually releasing it.